Actin forms a variety of well-known specialized structures such as lamellipodia, filapodia and stress fibers. But Sasha Bershadsky from the Weizmann Institute in Israel and the Mechanobiology Institute at the National University of Singapore is interested in a more mysterious but no less important actin-based network that appears to maintain cell shape.
The whole work began from observations made in laboratory of Mike Sheets, who showed that inhibition of myosin activity produces very weird morphology of cells, some very strange, unusual processes and holes in the cells, which shows that some actomyosin network probably is responsible for maintenance of cell shape. But about the structure of this network, still not much is known. So together with Mike, we come to the idea that we should study this actomyosin matrix.
To study this cellular actin network, Bershadsky and colleagues, led by grad student Wei Lo, plated cells onto cover slips micropatterned with adhesive strips of fibronectin. In the intervening non-adhesive regions, cells formed broad cytoplasmic bridges that contain the actomyosin matrix but lacked specialized actin structures involved in cell adhesion. The matrix was easiest to see when cells were treated with low doses of the drug latrunculin A, which removed other actin structures such as stress fibers.
If you treat cells with small doses of latrunculin, you begin to see the structures in the cell, multiple nodes, relatively uniformly distributed over the whole cell.
These actin nodes are more prominent in the presence of latrunculin, but they likely exist in untreated cells too. Super-resolution microscopy, for example, reveals similarly spaced actin nodes in between the stress fibers of untreated cells.
They spent a lot of time and skill to follow the dynamics of these nodes, and we found that they are very rapidly moving in the cell in a random fashion, but in fact not absolutely random. It is so-called drift diffusion. So their movement is a little bit biased because they are moving a little bit directionally.
Nodes within a few microns of one another tended to move toward each other and fuse to form a larger node, though nodes sometimes split apart as well. Bershadsky and colleagues then tried to identify other proteins associated with this nodal actin network.
Of course, we looked on myosin, and we also saw nodes, but myosin nodes, at least at the first several minutes of observations, were not coincide with these actin nodes, but were rather between them. And only after some time, myosin colocalized with these actin nodes. When we inhibit myosin 2 activity, either by chemical inhibitors or by knockdown of myosin 2A, these nodes actually stop their movement. So the movement is myosin-driven.
Lo et al. Then looked for other proteins associated with the nodal actomyosin network and found that the actin-nucleating formin protein DAM1 concentrated at actin nodes in the presence of latrunculin A and was found in similar patches in untreated cells too. In addition, the actin crosslinker filamin A was also associated with actin nodes in latrunculin-treated cells.
When we inhibit formin activity, again, either with a chemical inhibitor or by knockdown of this DAM1 formin, we noticed also that these nodes are moving much more slowly. It means that not only myosin contractility, but also polymerization of actin filaments playing a role in their movement. And finally, what about filamin? In the cells lacking filamin A, these nodes hardly formed. Obviously, filamin cross-linking is very important for formation of these nodes. But those nodes that still remain in filamin-lacking cells, they are moving actually faster than in control.
On the other hand, cells overexpressing filamin formed prominent but largely immobile actin nodes in the presence of latrunculin A, indicating that filamin is involved in maintaining and stabilizing the nodal actin network.
Altogether, this data led us to a model according to which these nodes are actually asters of actin. So actin filaments are growing from the formin down-one patches. So the plus end of actin filament are inside the nodes and minus ends are protruding outside. Filamin is also there, keeping all this aster-like structure together. But where is the myosin? Myosin initially is in between these asters, interacting with these protruding minus ends of actin filaments. But then myosin is moving these actin filaments, and this leads to movement of these nodes towards each other. In such process of fusion, myosin finally appear also in the center of these fused actin nodes.
Lowe et al. Developed and tested this model with the help of Alex McGillner's laboratory at the University of California at Davis.
Based on this simple model, Alex was able to make a simulation that very nicely reproduced the movement of these actin nodes that we observe. And it is additional proof that our idea about organization of these nodes was actually correct. In principle, such system is surprisingly robust, and in the cell, this matrix, we believe, is elastic network where fusion and fission of the nodes are in equilibrium, something that helps to resist the collapsing of the cell.
Even in untreated cells, then, this nodal actin network forms throughout the cytoplasm and may help cells maintain their shape. So what do Bershadsky and colleagues want to investigate next?
There are many interesting things that we can and we should study in more depth. First of all, we definitely should elucidate the molecular composition of this network. I will not be surprised if, in addition to DAM1, there are several other formins there, and there are many other important proteins that we may localize in these structures. Then, second question, we should, of course, look on dynamics of this network in more detail, not only in quote-unquote normal conditions, but how this network will change. In situations when cell experience different kind of mechanical stimulation. And last but not the least, we should try to see this network without latrunculin treatment. This, of course, was very convenient, but we are going to work using super-resolution microscopy to visualize it without this treatment.
In the meantime, you can learn more about the local organization and dynamics of cellular actin networks in the paper by Lowe et al., published in the September 30th, 2013 issue of the Journal of Cell Biology.